So this meeting is now being recorded, so thank you. Very warm welcome to everyone who's joined us for the How Can Biochemical Engineering Help Address Current Global Challenges webinar. So I'm just about to hand over to Alex. Alex will introduce the other members of the panel. I'm going to turn my camera off to save on bandwidth, but I'll be here and I might answer questions in written form with links as we go throughout the event. There will also be a survey at the end and we'd really appreciate your feedback. So Alex, over to you.
Thank you very much, Kim, and thank you everyone for joining. My name is Alex Kiparisides, and I am the admissions tutor and program director for the MSc Biochemical Engineering. And I'll just start straight away with a quick overview of how the conversation will flow today. I'll start by introducing the panel members, and then we'll discuss 4 key challenges. One being revolving around rapid response to epidemics and pandemics and vaccine manufacturing, so from the manufacturing point of view. The second one being talking around personalized and targeted healthcare manufacturing and the challenges therein. The third one will be around more environmentally friendly bioprocessing and materials. And finally, we'll talk about how learning and education may look like considering the circumstances we're currently all experiencing. So I'm joined today with Professor Martina Micheletti, who is the departmental tutor in the Department of Biochemical Engineering and the director of the VaxHub. Professor Gary Lai, who is the head of Department of Biochemical Engineering. Professor Paul Dalby, who is the co-director of the Future Targeted Healthcare Manufacturing Hub. So I'll pass on to Martina now to start introducing and discussing and start the discussion around the first global challenge. So Martina.
Thank you, Alex. Welcome everybody. It's a pleasure to come and talk about this quite a big challenge and a very topical one today. So I just want to start with maybe just giving some numbers regarding vaccines manufacturing, and then I can talk a little bit more about what we as a department, what we are doing to address some of these challenges. To make new vaccines, if you look at literature, previous examples, takes a long time, minimum 5 years up to 18 years. So years is really the thing to remember there. With usually requirements for very large budgets, so hundreds of millions dollars. Well, this information that are published is also a very, very complex industry. There is an inherent complexity of the material and the methods used. It is also something that commonly generates quite low revenues because vaccines are needed in specific countries, low and middle income countries. And they are needed for epidemic. And because it takes so long to make a vaccines which is safe and efficacious, it is often a risk to embark on the manufacturing, because by the time the vaccine is available, the epidemic threat might not be there anymore. So a very, very complex industry that is involved here. So if you think about what has happened in the in the last few months in the world, we went from the publication, I think, of the genome of the virus, I think early in January. We have now, so some of the vaccines, there are probably more than 150, around 170 vaccines in current preparation trial, at different stages of trials. We are now talking about months rather than years, so an incredible speed of response. Despite that, there are still some challenges, and I think in the UK, but also in other countries, I think the pandemic really showed that we were not prepared, and that and then there's a lot that we can do to improve our manufacturing capacities in this space. What are the areas and what are the possible solutions there? I'll talk in a second about what we are doing as a department, but some of the solutions are to essentially to have methods that are flexible, that are more generic, and they're scalable. Because if we have an incredible vaccine technologies that's safe and efficacious, but it cannot be scaled and made very quickly in very large number of doses, then is real use, is that effective? We need the solutions to manufacture more rapidly. And why is that? It's not only about the vaccine, which needs to be safe and efficacious, but it's also the actual process. The actual process actually is quite risky and can also fail. And we want to avoid that and have a very early data and rapid solutions. And we definitely need better diagnostics. And lately in the department, we have some ongoing research on that as well. And the final point is also that these vaccines and also the therapeutics are, needs to be accessible to countries where these are most needed. And with that, I mean, we need to make, we need to improve the sort of the types of agreement that we have. But one thing that we have done in the department is also to work on translation, on not only on technology transfer, but also on really translation of the technologies, and improve the learning and expertise in those countries where this is actually needed. I just want to maybe spend 1 minute on, next slide please, Alex, on what in the department, what are we, what do we have, and in terms of research, we lead something that's called VaxHub. There are different, you see on the diagram on top right, there's different institutions. This is a co-led by us with Professor Sarah Gilbert from the Jenner Institute in Oxford. You might have heard about her in the news, as she's really leading the activity on one of the leading UK vaccines. And what are we doing with this is really, there are a number of researchers in Oxford and UCL and in some of the other spokes that work on developing vaccines, scaling this up and finding manufacturing solutions with a specific benefit and remit to low and middle income countries, ensuring that these vaccines are, that we ensure that we are securing essentially this supply. And we are also translating the technology and the learning to the manufacturers for local, to support local manufacturing approaches. We work on different types of vaccines, and you see there's lots of different users group with the small and large companies, and all this sort of body of research is also translated in some of our teaching programs, in the teaching program, via sort of, topical research projects, but also on, to develop new new learning material. So I don't know if the other panel members would like to add anything to these slides or comment on some of the issues that I've raised.
Yeah, thank you very much, Martina. So obviously, from a manufacturing point of view, what would be the major challenge? So we have, several vaccines that are being trialed and tested, and probably a small amount of material is generated for the clinical trials. And this is potentially a successful vaccine would be needed in enormous doses around the world. So what would be the key challenges, and maybe what are the possible technologies that would allow us to respond?
I think that in terms of challenges, specific challenges would depend really on the actual method. So some of the, so for example, the viral vectored vaccines is a relatively new technology that now seems, definitely really promising. But the problem and the main challenge there was that, we had back in January, we had some sort of process to make a vaccine of that type. but it was not optimized. So for example, we had very small number of doses from, liters and liters of starting material. So of course, that means that, we needed very, very large bioreactor, and the capacity in countries might be actually limited. So essentially there were challenges at each process stage, which really needed to be optimized. So I can maybe make an example of some of the technologies that we work on that could improve the sort of rapidity of manufacturing, the speed of manufacturing. So one of the things that the department has pioneered is the use of ultra scale down tools. What are these? And so these are sort of small scale devices that mimic a specific critical stage of the large scale process. So if you think about your material as it goes through the process, what it experiences at the large scale is usually very different to what it experiences when we do the same process at the bench scale in the laboratory. So this tool are able to sort of mimic large-scale operations, and they've been very popular in with our sort of industry industrial collaborators, because they're able to mimic how sort of essentially what the material experience at large scale in order to get early manufacturing, in order to get manufacturing in site very, very early on, and essentially de-risk manufacturing and predict and limit the degree of failures.
Okay, so it's really not. There's multiple challenges, right? There's a huge global demand. There is a huge uncertainty regarding the technology of the vaccine, so the form and the properties of the final pharmaceutical product, let's say. Therefore, process development cannot really truly start in parallel as you would normally do, let's say, on a platform process, but you sort of have to wait and see which one of your candidates will sort of break through the clinical trials.
Actually, I think that many sort of review publications actually in the vaccine space suggest that, especially in the vaccine, for vaccine processes in general, not even in a pandemic scenario, that the actual process development and manufacturing is started very, very early on. And I think that this would be even more true for a pandemic scenario, because if you think about, once the clinical trials are, they show that if essentially the vaccine is safe and is efficacious, then, it needs to be started manufacturing, but there is a lot of time spent on sort of meeting regulatory requirements, and it's not only the molecule which is sort of regulated, but it's also the process. So you don't want to change process. So you want to have very confidence in your process very early on, in order not to make changes later that essentially could kickstart a requirement to do trials again. And that's another, quite a significant risk in the vaccine space.
Yes, Paul?
You have a slightly unique scenario under pandemic where you're not just a company trying to develop a drug, you've got government money coming in and large amounts of it. So actually a risk can be taken to create the manufacturing space before you even know the clinical trials will be successful. For a company on its own, they can't do that. That's too risky. It will cost them money. So this is very different.
Can I just add, or ask Martina to comment on what they're doing within the VaxHub to address vaccine to supply to developing countries? So as well as some sort of safety and regulatory concerns, I think there's also moral concerns that we have about how do you make these vaccines available to all. So you have some extremes, like we recently seen the US government has bought up the whole of the supply of Gilead, one of their therapeutics, for the next 2 years. How do you address within the VaxHub these issues of global supply of vaccines, particularly developing countries?
Yeah, so there is a few things that we do in the hub. I think one aspect which I'd like to mention is the research effort to, maybe a couple of things. So one is definitely the research effort to find solutions that make the vaccine production itself much cheaper. I mean, the minimum cost for a manufacturer of a vaccine is the cost of production, right? So, and obviously there's a lots of challenges in doing that, but essentially, so this is why we need really innovative solutions. So, one of the things that we're working on in that space, if people might be familiar with the vaccine marketed by Pfizer, Prevnar, is quite expensive. What we are trying to do is to find an alternative which is much simpler to make, using, there's a conjugation process which can happen, instead of being made synthetically, can be, in vivo within a cell. So essentially we are exploiting, cells as factories in order to make the sort of conjugate that acts essentially as a vaccine. So and that is one of the sort of research of the efforts. And another thing which is important to do in that we are trying to do in the VaxHub is really contributing to is really working with some of these companies. We establish long-term relationship. We try to understand where the issues in their manufacturing processes are, and we can, so that we can sort of work together on resolving those. And there's a number of workshops that we are organizing, but which essentially including support for example, for online courses on some of these sort of new technologies that would not be otherwise available to some of their employees.
Okay, thank you very much. We have a couple of questions in the chat, but let's try to revisit them at the end, and let's move on to discuss a bit about our next challenge, which is personalized healthcare, and obviously we're going to be talking about this from the challenges from a manufacturing point of view, because it's a radically different way to approach medicine manufacturing. So Paul, if you could kindly introduce the area to us.
Yeah, so this is work that's ongoing in this Future Targeted Healthcare Manufacturing Hub you can see on over my shoulder. So this is similar to the VaxHub. It's a collaborative with 5 other universities across the UK and 35 companies in biologics manufacturing. So they're making cell therapies, gene therapies, mRNA therapy types, they're making proteins, antibodies, etc. The vision here, as you can see right at the bottom, was that healthcare is gradually becoming more and more personalized. And what that means is, at the moment, you would put in, say, 10,000 people into a clinical trial, but know nothing about those people. And so maybe 3/4 of them don't respond to your drug candidate. But that might be because they were genetically not predisposed to respond anyway, right? So their disease might have been a slightly different version of the disease. And so what's happening now is, particularly in cancer, so oncology, is they are sequencing the cancer and understanding the different genetic variants in those cancers. And then you can target a particular blend of therapies towards that patient. So that's one option. The other is what's basically where you take cells from the patient, and this is the CAR T therapies, and you sort of boost them into a specialist cell that can go and find the cancer and attack it. And that's like the CAR T, for example. So you genetically engineer somebody's own cells and put them back in. So that's very personal. So it's made out of someone's own cells. So both of those kind of approaches are emerging. And the question is, how do we deal with that in a manufacturing sense? So similar to the vaccine story, speed is of the essence, but also now potentially manufacturing very close to the patient. So there's talk now of decentralizing manufacturing. So instead of an offshore large scale manufacturing plant that makes the medicine for everybody in the world, you could now talk about small manufacturing boxes even, so-called factories in a box, which are housed in hospitals and clinics. And when somebody turns up with a particular genetic variant of a disease, which the sequencing can be done in hours to find out, then you can go and put the relevant bits of genetics into the machine, and it will be like a printer, and it will turn around and give you your medicine back. And so that's on site, it's ready to go. And it means medicines can be a lot less stable, for example, so because they're not going to hang around, so you don't have to worry about storing them for 2 years, for example. So all of it's changing towards this bedside manufacturing. To get there, the technology has to all change. The ability to do manufacturing in real time, to monitor the process, to qualify that the product is safe before you put it into the patient is all very difficult when your dose that's coming out of the box is for one person. There's not, like, you know, in a traditional manufacturing plant, you might take 1 in 10,000 samples and make sure that they're correct. You can't do that here. So you have to have the process absolutely robust, so the product is always the same. So that's the kind of sort of vision of where manufacturing is going. So it's real just-in-time manufacture. It's made to order. Okay, that's just
okay.
Open-ended from there, I think.
Okay, thank you, Paul. Thank you very much. This, you know, to someone maybe coming from the outside, this all sounds a bit futuristic, perhaps. Do we have any sort of at-scale demonstrations of the concepts of this technology working? Are there?
Of course, yeah. So examples would be Yescarta, Kymriah. So these cell therapies. There's now just approved from Roche. There's a dual antibody therapy which is targeted to certain cancers. So they are emerging. The challenge is that because they're the first of their kind, they haven't really worried about making them as low a price as possible, a low cost of manufacturing as possible. And they cost on the orders of $500,000 upwards, some ridiculous prices for some of them, which clearly, as a first mover, you could maybe get away with that. But as more and more therapies come in based on the similar technologies, there isn't the room for them because the people supplying the money into the system, the healthcare providers, can only afford maybe 1 or 2 at that price. So the challenge now is to bring their price down and make them more accessible.
Yeah, so just to get the conversation going around this, so we've been using cells to produce things for many years now, so it's nothing new. Why are these cell-based therapies so much more challenging? And it almost seems like we need to reinvent the wheel in some ways, as in process, or the manufacturing process is rather archaic, let's say, in a way, compared to some of the other bioprocesses or biomanufacturing processes.
So I think the first thing is to understand the time scale. Right? So this is made out of your own cell, and then you're engineering it. That's actually fairly unique. We've been engineering E. coli and yeast cells for millennia in a way. But you know, in the last 30, 40 years, seriously, for biotechnology. Now we're talking about taking a cell out of your own body and modifying it genetically. And you've got somebody in the hospital, you've just taken their sample, and you want to get the therapy back to them while they're still in the hospital. And the turnaround time for that is estimated to be typically running at something like 20 to 30 days. They'd like it to be more like 14-day turnaround, so the patient's not there for too long. So that's one challenge, is kind of doing that. The other is to make sure only the right sample comes back to the patient. And the other is to make sure it is actually what it's supposed to be. And the analytics around that are phenomenally complex, because there's a whole cell, and they look very similar to the cell that first came out of the patient. It's very difficult to know whether you've now got an activated, as it's called, cell. And we don't really have the foolproof analytics for that. We have so-called panel of markers, which are strongly suggestive that it is correct. But this is all territory that needs to improve as these therapies develop.
Yeah, so it really is very, very challenging from a manufacturing point of view, because you sort of lose a lot of the benefits of large scale manufacturing, and you introduce a whole other host of new challenges, because you want to do it A, rapidly, B, almost surgically in terms of accuracy and quality of the final product with limited analytics.
There's one other critical issue, and that is, you know, if you're doing a process using E. coli, you know that E. coli, you know it well. So that batch, each time you make a batch, it will work the same way unless you've done something wrong. Now you have cells coming from different people of different healthy status. They've got cancers, they've got immune suppression going on from previous therapies. The cells that are coming out of them are not necessarily in good, healthy state, and the variability is huge. So how do you make a consistent product from their cells when their cells are, as a starting point, are already very different? That's a big, big challenge.
Okay.
Can I ask one quick? So just wanted to explore this issue of cost a little bit more. So, you know, Paul mentioned that these patients are already in hospital. So they're already pretty critically ill at the point that you start to harvest the cells and do your genetic modification. And Paul mentioned, you know, the costs of $250,000 per treatment. So how do you weigh up the, you know, what's the appropriate cost for the therapy? Because there's clearly difficulties in the sort of manufacturing, but then there are advantages in the sense that these are revolutionary medicines, in the sense that they actually cure the disease rather than treating the symptoms like, say, a transplant might be. And therefore, you've got all these potential savings in future healthcare costs that you're not going to be susceptible to if you use the T-cell therapy. So how is the pricing of these medicines arrived at?
Yeah, so we've worked closely, obviously, with industry itself, but also with the NHS trusts, NHS England, NICE, which is the National Institute of Clinical Excellence, policymakers and health economists. We get all of those in a room, and we've had these sorts of discussions. So the costs saved are time in bed for a patient receiving other treatments that aren't going to work, right? So if we can cut that out, you can make enormous gains. So, you know, interventions that just basically keep a patient in the system, that all costs money as well. So this is now a one shot, does the job, out you go, don't come back. And so it should lower the total cost of healthcare, even if the actual therapy itself at that particular one time is on the order of about 10 times higher it is, roughly, right, for a typical drug. Now, at the moment, that doesn't quite work because it's such a new therapy, it's the last stage, it's the last resort. So they have actually already had all those other interventions, and it's when they don't work that they now get this therapy, which can rather sort of strangely means their cells are in the least capable position to have that therapy. So it would actually work a lot better if they could be brought earlier, and that's currently where it's moving. So as trust grows in these types of therapy, they will move further and further up towards being the first barrier. And then it doesn't matter if it costs $500,000, it will save more money than that in terms of other administrative healthcare costs.
Okay, great. Thank you very much. We have a couple of questions in the chat, but we'll move on to discuss some of our other challenges and keep up with time. So our early discussions has really been focused about around healthcare and biomanufacturing. That's obviously a very big avenue and career sort of target for many biochemical engineers. But the contributions of biochemical engineering do not stop with medicines. There are many other areas and also equally important. And Gary might help us introduce some of the contributions of biochemical engineering towards a more sustainable future.
Okay, thanks, Alex. You stole my introduction there. But yeah, so clearly the previous examples have been very much healthcare focused, but biochemical engineering skills, and it perhaps links to what we're going to talk about in Challenge 4, the sort of educational aspects, the skills that we teach and learn as biochemical engineers can be applied to lots of different challenges, and one of those is in the area of sustainability and the environment. Perhaps at the current time, under the COVID situation, we see that, you know, there's obviously a pandemic and there's a international emergency. But if you're sort of following the sort of exploits of Greta Thunberg and what she's been saying to promote the environment, you'll have seen that in the last few weeks, she's changed her tone to talking about this now being a climate change emergency. And I think that's balancing up the focus of where government efforts need to be, not just in the healthcare, but also in the environmental aspects. And that's something within what we do in biochemical engineering at UCL, and particularly around trying to address how we can engineer a more sustainable future. And this is perhaps one of the key areas that kind of links with developments we're doing in biocatalysis, synthetic biology, with the process engineering work that we do as well. So the root cause is that we are heavily addicted to petrochemicals. And in our modern society, we rely on having lots of stuff around. And the problem is that all that stuff that we rely on is made from petrochemical resources. So I put a list of 3 here on this slide. So, you know, if you think about the fuels that underpin our transport, about our heating, they're all from petrochemicals. If you think about the materials that we use, the plastics, the polyesters, the things we use in our clothing, these are all derived from petrochemical resources. And probably less well known is pharmaceuticals. So if you're thinking about synthesizing your conventional chemical pharmaceutical, then they're either reliant on intermediate molecules that are derived from petrochemical resources, or they're using organic solvents, which are again from petrochemical resources. So all these things that are now critical parts of our modern society, we can all trace them back to petrochemical sources. And then we have the environmental issues of sustainability, CO2 emissions, and climate change. So there is an emergency of how do we get away from our addictions on petrochemicals. And that's one of the things we focus on in the department, which is looking at, are there alternative and renewable resources that will allow us to manufacture the fuels, the materials, the pharmaceuticals that we need. So this is challenging for similar reasons to how we talked about the manufacture of some of the more advanced medicines and the vaccines. In that we have to do it in ways that are themselves environmentally friendly, but also very, very cost effective. And we have to come up with solutions which are holistic. So if we're thinking about taking a particular crop, then we have to use all the parts of that crop in order to come up with a total solution that is still environmentally friendly. And I put an example on this slide of one of the projects we were involved in recent years, which is around the conversion of sugar beet, and particularly sugar beet pulp, into these kind of fuels and pharmaceuticals. So the top image that you can see is sugar beet that's harvested from the fields. And the image at the bottom with the person holding the hands is the sugar beet pulp. And this is the waste material that's left after the sucrose has been extracted out of the sugar beet to make your table sugar. And why were we interested in sugar beet? So one of the key priorities here is that if you're going to use a renewable feedstock, you don't want to have to transport it around the globe, because that uses up fuels and contributes to CO2 emissions. So you have to find solutions that are local for particular countries or regions. And the UK and Northern Europe has a very large annual sugar beet crop. So we have about 80 million tons of sugar beet produced in the UK per annum. And the beauty is that it's regionalized, and it's all taken to sort of centralized biorefineries. So you don't have all your transport and CO2 emissions related to transport. So this is a good crop for the UK. Out of that 80 million tons of sugar beet that's harvested, we currently produce about 200,000 tons of the sugar beet pulp, and that's normally either goes to waste or is used as an animal food supplement. So it's that sugar beet pulp that we've been working with. So what do we do with it? We basically have to come up with solutions that use all the material that is present in that sugar beet pulp. And in the case of sugar beet pulp in particular, the main components are cellulose, which is a polymer of glucose, and the other component is mainly pectin, sugar beet pectin, which is a polymer of galactronic acid, galactose, and arabinose. So these are other types of sugars. So if we're going to use the sugar beet pulp as a renewable feedstock, we need to process it, to break it down to release these polymers, and then think what we can do with the sugars that are released. So in one of our research projects, what we showed was that we could take the sugar beet pulp, we could do a sort of mild heat treatment that would release the pectin and separate it from the cellulose. And then we showed that we could take some enzymes that could break down cellulose into glucose. And then that glucose we could feed into a yeast that could then grow on that in order to make bioethanol. And then that bioethanol could be used as a food replacement. And that was a very efficient process. So about 100% of the cellulose we could break down to glucose and get quantitative conversion into bioethanol. So that left us with the pectin fraction. And what we showed there was that, again, we could use some enzymes that we cloned from nature. We could selectively release the arabinose, which is one of the components of the pectin. And then using other enzymes, we could modify that to make it into much higher value pharmaceutical intermediates that could be used to synthesize a range of medicinal compounds. And then the remaining galactronic acid, what we showed is that, again, we could use other types of enzymes in order to start to polymerize the galactronic acid into polymers, which could then make biodegradable polymers to replace petrochemical derived polymers. And so what we came up with was really a solution where we could take the whole of this waste stream, fractionate it into a range of different components, and then convert each one of those into one of these useful products that will replace petrochemicals and therefore contribute to allowing us to keep our current standards of living, but at the same time reducing our reliance on petrochemicals and improving the environment. So that was a successful project, and one of the things we're doing now is we've recently submitted another grant to look at sort of scaling up and looking at the sort of commercialization of some of those technologies.
Okay, thank you very much, Gary. That was a very thorough, very interesting overview of some of the possibilities that what we can achieve with biochemical engineering. And I know I've read people are trying to sort of apply similar concepts to all other types of waste. There was something recently in the news about ground coffee from all the cafeterias around the world, and that's a huge waste stream that has a lot of value still in it. Now, what I found interesting is that in this process that you described, almost with this biorefinery type of approach, it was not just the cells, which intuitively a lot of people might think, yeah, okay, we we find special cells, or we engineer special cells, and we throw them in, and stuff happens. There's actually the enzymes, and that sort of, I guess, opens up a lot more opportunities, because there's a huge variety of enzymes in nature that can be used to do all sorts of useful convergence in an environmentally friendly way. So maybe, I don't know if Paul could answer this. How challenging is it to identify an enzyme that does something interesting, that performs a conversion that is of use to you, and sort of industrialize it, if you wish, so use it in an industrial type application?
Well, all right. So obviously it can be done, and it is being done, at an industrial level. People are discovering lots of new enzymes, but to say it's easy would be misleading. I think it comes down to a lot of factors. One is, is the reaction you're looking for feasible in terms of the chemistry in the first place? Is it likely that an enzyme could have done that? And that, most chemists will be able to kind of look at a chemical reaction and tell you whether that's thermodynamically impossible or just, unlikely to be catalyzed. The next question is, can you create, because to find a new enzyme, it means screening usually thousands and thousands of possibilities. And you can do that increasingly by computational methods and bioinformatics based on known enzymes and what they do. But by and large, on the ground, it's done by physically screening whether something catalyzes your reaction or not. So you need to initially have a screen in place that can be used in automated high throughput sense to, quickly tell you which samples have got an enzyme in there that does your conversion. If you have that in place, then it is about just setting up your screen, so accessing lots of enzymes. And on the one hand, there are lots of commercial kits now of lots of variety of enzymes present in them, but that is still fairly limited in the overall space that is available within the enzyme, overall space, if you like, because there are billions and billions of possible enzymes out there, of which we've kind of scratched the surface and taken, on the order of thousands out, probably to millions at most. And most of those are similar or the same. So there's a long way to go if you've got something quite exotic that you want to do that has no near precedence. There's a long way to go to make that easy to get something. A program of research, 6 months, a year, 2 years, you'd expect to get something out that might work. And then when you get that, it might not work at scale, or it might not work in particular conditions you need it to, or the process intensity you want it to work at. So there's still some more work to do. So yeah.
And then obviously I think once you've identified an enzyme that does the trick, even if it can't do it at economically or a rate that makes it economically viable. I suspect a lot of our listeners have noticed the Nobel Prize for Chemistry for Francis Arnold last year around this technology called directed enzyme evolution. And so that's something that Paul and other colleagues are looking at in the department, of once you've identified the enzymes, actually improving their activity and their stability to make them robust enough to use in these types of large-scale industrial processes. I noticed there was a question in the chat, which was 238 anonymous attendee. I don't think that's their real name, but just to try and address it. So the question was, regarding the production of these renewable sort of resources, there's different paths like sugar beet pulp, wood chip pulp, and are these cost effective and realistic to pursue compared to regular sources? I think there's two parts to that. One is the sort of more philosophical part, which is, are they realistic to pursue compared to regular resources? I think yes is the answer. I mean, we have been reliant on petrochemicals, but it's at the detriment of our planet and our environment. And so I think we have as scientists and engineers, a sort of duty to pursue these alternatives and show that we can make the types of products and the fuels that we're interested from them in a renewable way. The difficulty actually, I think, is the first part of the question, which is, can we do it cost effectively? And that's really whereby there's lots of different potential sources of renewable feedstocks. Many are geographically or country specific, and actually there's lots of technologies available for their breakdown and conversion into the products that we need. The challenge is really that cost of how you put those technologies together and do it in a way that's cost effective as well as environmentally sustainable. I think that's the challenge. We need to use our process engineering insight to think about how we design these processes and we intensify them. And then we've also got to use some of our biological insights of applying some techniques like enzyme evolution, genetic engineering, in order to make the biology work at these kind of intensities that we need it to. And so it is possible, but there's still lots of challenges and research to do.
Okay, thank you very much, Gary. And now let's move on to the next challenge, which is, I guess, burning in everyone's mind. How will education and specifically the teaching and learning experience at UCL look like for the coming academic year? And perhaps Martina would like to make an introduction, and then we can all chip in on how things will look like starting from September.
Yeah, okay, so I have the biggest challenge here, I think, of all. So this is, yeah, it is very difficult, right? Because we have, for years, we have had a model where we were teaching, in a classroom, with looking at our students, interacting with them, UCL is a London university, is a global university, but it's a London one, and this was the model that we have operated for years, and it's quite a big, it requires quite a big shift now to think in a different way about, the teaching. We had some experience, we started some experience with our current students in the last few months, but it is now about, the preparation for the next term and how we can really provide a similar experience, despite the limitations, which, the government regulations on social distancing and other limitations which, are imposed by, by such a big institutions like UCL, and travel limitations, travel restrictions as well. So can start by maybe providing some information on what we are thinking. It is very important, even if we, provide some material and teaching online, that we foster a sense of community. And we are a smallish kind of departments, and our students benefit from, some sort of bespoke support, but also a lot from, from interacting with the community of staff and of other students, which can be doctoral students working on different projects like we have just talked about. And so, in terms of this is what really makes the experience at UCL quite rich. So we are trying to find ways in which we can provide a similar experience and this type of interaction, with the new cohort, and do cohort building activities, but also how you can sort of interact, with the staff and with our students.
Sorry, Martina, just maybe we should make a clarification here in case, not all prospective students are keeping up to date with their emails and UCL communications. But the reason we're discussing this is that our traditional lecture theater type of teaching will be held online for term 1 at least. While there will be some face-to-face activities happening in London, if in term 1 these will be very limited in number, and this is due to the restrictions imposed by social distancing measures. I'd say the majority of the learning experience for term 1 will be relying on some form of online content delivery, and it is what we are discussing now. How do we design? How do we replicate the benefits of a London-based educational experience in a department that's known for being tightly knit and having very direct communication between a very good student to staff ratio? How do we replicate that experience online?
Yeah, no, thank you, Alex. Yeah, I thought I was working on the basis of, previous.
Yeah, I'm assuming everyone knows, but just in case there is someone sort of.
Okay, thank you. So yeah, just one more minute probably for me. So we're trying to work on the material and repurpose it and use it to make it a little bit more, to make it more engaging, but suited to a different type of provision. I don't think that there's not everything is negative. I think about the online approach. I teach MSc students from very different backgrounds, and the ability to go through the material, or at least some of the material, in your own time, rather than in a very sort of restricted timing before a lecture, is or even during a lecture, is something that I think some of my current students would really appreciate. Just because of the wide variety of learning approaches, but also the variety of backgrounds. And so I hope that this is one of the benefit of our students next term. Big question on how do we have the assessment, current regulations, social distancing, etc., would make it very difficult or impossible to do invigilation, to do appropriate invigilation, and maintaining, obviously, all our courses are accredited by the Institution of Chemical Engineers, and therefore we have responsibility to ensure that such requirements are met. And this is another challenge for us. I'm just going to stop here. I'm just concerned about time, so I just want to leave space for others to chip in if they want to, and for questions.
Yeah, so we already have a question related to this, which is how will laboratory practicals and lab sessions work in term 1? Now, I can answer this for the MSc, and then maybe Martina, you can also add some thoughts at the end. By design, the practicals for the MSc are timetabled for term 2. So we had no practicals in term 1. We had bunched them up all in term 2 by design. In addition to that, to sort of mitigate as much risk as we possibly can, we're trying to create space in the timetable in term 3, so we can move practical to term 3 if we need to. So we have done a thorough review of all our practical sessions, and we are now creating the space in the timetable for term 3. Our hope is that they will happen normally and naturally in term 2 as they have been originally timetabled. We sort of have a backup plan. Okay. So I guess an interesting point is we were all, online teaching, full online teaching of the program was a bit unknown to us when we had to do it one Monday, where we woke up and said, okay, now you can no longer go into the lecture theatre, you have to do this online. But I found it refreshing in the sense that I got a lot more engagement from my students online than I would get in a normal lecture theatre. Whenever I opened the virtual floor for questions, I would get a lot more questions in my chat box than I would get if people had to raise their hands and and speak in front of the entire lecture theatre. So I found that refreshing from my point of view. I don't know what thoughts other members of the panel have on this experience from the tail end of term 2.
And the other experience is obviously the current MSc students are still going. They're doing their sort of design projects over the summer. We've just run our first week of design using a Teams-based format with lots of industrial input, both to the delivery and the assessment. And the initial is actually our really pilot for how we'll be teaching, certainly the first term in the next academic year. And the feedback has been really good, actually. So you know, we've had industrialists who teach on a lot of our programs have pre-recorded their lectures and videoed them and delivered them. They've come and helped us with the assessment. So we've set up teams or groups within the Microsoft Teams environment or through things like Zoom. And there's been a lot of interaction. So I think it's actually, there's a lot of discussion around what the new normal will be post-COVID. But I think it's, as a department, with my head of department hat on, it's made us look at a lot of these new teaching methods and really explore them. And I think what will come out with the other side is a slightly different and rebalanced approach to teaching, which is, you know, partly live synchronous together in the lecture theatre, and partly remote asynchronous, where people have the materials available to, you know, high quality materials to study themselves. And then just say the other thing we're investing in is a range of software packages that allow us to help out, particularly on the practical side. So there's a lot of good laboratory simulations out there that we're using to prep people so that when they do come into the laboratory, they maximize their use of hands-on resources. So I think it's going to be a really interesting time next year, very much exploring this, I think, what will be the future of educational delivery, not just at UCL, but I think globally.
Okay, thank you very much, Gary. And again, it's important to note that campus will be open from September to welcome new students. Obviously, there will be social distancing on campus. There is a one-way system that has been introduced into campus to avoid people coming or walking across each other, and to minimize the risk. And I believe most buildings will work at a reduced capacity, and the priority will be given to students. So staff members, where possible, will continue avoiding campus to allow students to make use of the centralized facilities such as the library and the student center. Now, on top of all the activities that we're designing online and planning for, there will be face-to-face activities happening throughout the years for the students that do make it to London. And these will be what we call internally enrichment activities. They will not be sort of connected to the assessment in any way, but they will be designed to enrich your experience and really provide that campus-based feeling.
Hi, I'm just going to quickly, whilst Alex is going through that, point out that we've also got some links to the undergraduate and postgraduate events committees posts on the student room. Alex is also presenting a Q&A session on the 21st if you've got any more questions about the MSc programme. And I've put a link to a survey to ask for your responses from this webinar. So if you get a few moments, the links will be up there for the next couple of minutes. We're coming up to, in fact, we're 3 o'clock. So Alex, do you want to, any last questions that you want to go through?
There was one question. So just quickly, there were a couple of questions regarding admissions, swapping from BEng to MEng and stuff like that. If you see on your screen the email b.admissions@ucl.ac.uk, you can email those types of questions directly there. Anything related to the MSc, you can use the email on top of that, which is my personal email. And as Kim mentioned, we'll have a dedicated just Q&A session, no presentation, just me answering your questions live with anything related to the MSc. So with that, I'd like to thank all our panel members for their thoughts and inputs today. Kim for organizing, and all of you for attending and really engaging with the discussions and having a lot of questions. I know there are a lot of questions that have not been answered yet. We can sort of try to answer them later on, or you can send us an email and we can try to reply to your questions. So yeah, thank you very much.
Thank you very much for joining. As always, please get in touch with us if you've got anything afterwards. So thanks very much.
Bye-bye.
Thank you. Bye-bye.
Bye.